Perfect. All right, well, hello everyone. Thank you for joining us, and sorry for the delay again. We're just having a few issues with WebEx, but I think it's safe to say we'll try something else for next week. Thank you for joining in on us for the first seminar of the year. We are the Marine Institute Graduate Society, and we put on a seminar series throughout the fall and winter semesters. We meet typically every other week on Thursdays at noon. And this is being recorded and will be uploaded to our YouTube channel, where you can access all of our past seminars as well for the past few years. Before we start, we do want to provide a land acknowledgement. We respectfully acknowledge the land in which we gather as the ancestral homelands of the Beothuk and the island of Newfoundland as the unceded traditional territory of the Beothuk and the Mi'kmaq. We would also like to recognize the Inuit of Nunatsiavut and Nunatukavut, and the Innu of Nitassinan, and their ancestors as the original peoples of Labrador. We strive for respectful relationships with the peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. So today we will hear from Dr. Heather Koopman. Dr. Koopman is a marine physiologist who wears two hats. She is a professor and chair of the Department of Biology and Marine Biology at the University of North Carolina, Wilmington, and senior scientist at the Grand Whale and Seabird Research Station in New Brunswick. She has earned a BSc and MSc from the University of Guelph, a PhD from Duke University, and completed a postdoctoral fellowship at Woods Hole Oceanographic Institution. Her research interests include marine lipids, lobster movement patterns and reproduction, and the relationships between zooplankton quality and quantity and climate change. She has been a faculty member at UNCW since 2004, and a biologist at the Grand Whale and Seabird Research Station since 1991. She has more than 60 peer-reviewed publications and has secured more than $1.8 million in research funding at UNCW and $700,000 in support of the Grand Research Station. When not working on science, administration, or teaching, she enjoys baking pies and walking her dogs. And one of the dogs is actually sitting right next to her. That's Cooley. So with that, I will now pass it over to Dr. Koopman.
Okay, can everyone hear me?
Yep.
Okay, now I've never used WebEx, so this is going to be fun. I'm going to try and share my content here.
Yep, and apparently I just shared the page.
Okay, yeah, sorry, I have to, my system setting is now requiring a password. Sorry. Oh my goodness, my computer is not allowing me to share with you.
Of course not. The WebEx adventures continue. Maybe.
Oh my goodness, sorry about this.
Oh, it's all good. If you perhaps send us the presentation, we can share it from our screen.
It's huge. Hang on a second. I'm just trying to, so UNCW has these brilliant security settings where you're not allowed to change anything on your own computer. So hang on, I am now.
Yeah, we have the same problems here. Hence the issues this morning.
Okay, now let's see if this works. Oops. Oh my God. Okay, that's all fine. This should allow me to share. Okay, it's telling, I think I have to quit and go back. So let me get out and I'll come back. Hang on. Sorry about this.
Good luck.
Oh my God.
There you go.
Okay. All right. Okay, can you hear me?
Yes, we can.
Okay, and now if I do this, do you see?
And we can see the presentation, so it looks perfect.
Okay, I'm just going to start.
Okay, good luck.
I understand if anyone needs to leave early because we've had these technical problems. So I'm going to talk to you a little bit about the Bay of Fundy, zooplankton, lobsters, and water temperatures. So for those of you who are familiar with the Bay, it's got this reputation as having the largest tides in the world, which it does. So just to kind of orient you, we have Nova Scotia here, New Brunswick, hopefully you can see my pointer. And these numbers here is just denote the tides. We get high and low tide twice a day. And this blob here, which you'll see in a little bit more detail later, is Graminan Island, which is sort of my research home in the summertime and now also November. These high tides, or large tides, have incredible impacts on everything you do in the Bay of Fundy, from, for example, how you tie up your boat to what the weather looks like. It can be an absolutely lovely day with a little bit of a southwest wind on a flood tide, and then the tide turns to an ebb, and all of a sudden you're in a sea state 3 or 4, and in a small vessel, you're going to have to go home. This also does impact currents. You know, we get some ripping 4 or 5 knot currents through some parts of our study areas, and that can impact everything from how you're maneuvering your boat to what's happening with your plankton net. So there's a lot to think about when you're doing research in our neck of the woods. But because we have these enormous tides, we also have these regions of upwelling, which leads to incredible biodiversity. And this is just a snapshot of some of the animals. Maybe, let me see if this will go into presenter mode. Is that better? Yes. This leads to incredible biodiversity, and these are just some of the organisms that we've studied in the past and are currently doing so. And today, I'm going to focus on these guys over here, these little zooplankton, calanus, and lobsters, and we're going to throw in a little bit of basking sharks to give us some oceanography data, but I'll come to that at the end. So I'm going to tell you a little bit about the research station, not much since we're already a bit late. A bit about zooplankton, the important species there, Calanus finmarchicus, which I believe you also have where you are. A bit about lobster reproduction and working with the commercial fishery, and then some data on water temperatures, which are going to sound a little confounding, but they're really not. So again, here, this map on the left shows you the greater Gulf of Maine, as well as the Bay of Fundy up in here. And the red circle denotes Graminan Island, where the Graminan Whale and Seabird Research Station is. That's this house here. The Graminan Whale and Seabird Research Station was started in 1981 as a place for graduate students from the University of Guelph to come out and do research in the Bay. Since then, it has changed into a completely independent Canadian nonprofit organization run by a handful of us. We have a research station, we have a small museum, we have a little gift shop, and we have to raise all of our own money to do all of our own research. Some of these pictures here just show you a little bit about what we do. This inflatable down here, this is Xiphias, which is actually the Latin name for swordfish. This is one of our research vessels. The one on the upper right-hand corner here shows you one of the lobster boats that I work on, then some lobsters, and this is a picture of a basking shark that was doing a good job at following us this summer. Oops. So just to tell you a little bit about Calanus finmarchicus, this is a super important member of the zooplankton community. They're small, but they are mighty. We find them in most of the North Atlantic, so the Bay of Fundy and other northern seas. And the important thing about Calanus is that one of their life stages forms really dense swarms at the bottom when they're about to undergo diapause, and I'll talk about that in a little bit. But the big deal about this particular stage of Calanus, which I'll show you in a second, is that they have this oil sac because they're storing energy for a period of diapause where they're not going to be feeding for a number of months over the winter. And this gives them a really high energy content. And so they're an important food source for North Atlantic right whales and other animals. And I'm interested in lots of other animals, but I'll tell you, and one of my points at the end is being creative about how you get funding. People are interested in North Atlantic right whales, and so it's a way to sort of do a lot of interesting ecosystem studies when you tie them in. So why do we care about Calanus? This is the life cycle of Calanus fin marchicus. So they have 6 naupliar stages after the egg. Then they go into 6 copepodite or copepod stages. And the super important one is what we call C5 here, or the 5th stage, because this is the one that's going to overwinter at the bottom before they become mature adults and then reproduce. These are the guys that have the really high energy content because they have that oil sac, and that's why they're so important. You can kind of see the oil sac here in this image of a Calanus taking up a huge amount of space in the body. And this makes them really important for predators that feed on them directly, like basking sharks and North Atlantic right whales. And also indirectly, if you think about all these guys at the bottom, humpback whales, seals, and tuna all feed on herring, and herring feed on copepods. So they're really important for most aspects of the organisms in the Bay of Fundy and in other places. Here's a nice image that really shows you the oil sac of a Calanus, sort of enlarged here where you can see that. And a lot of other copepods and most other life stages for this species don't have that, that oil sac, which makes them, you know, worth more, I'll use my Canadianisms here, worth more of a fudgio than a piece of popcorn. So how do we collect the samples and where have we been doing this? This map on the right shows you again the lower Bay of Fundy. Here's Grand Manan Island. And these are the shipping lanes that go in and out towards St. John. And this blue blob in the middle is the conservation area where we have the highest concentrations historically of where we used to see North Atlantic right whales. And so we decided way back in 2006 that we were going to start collecting zooplankton samples in this area and monitor the zooplankton over time. And that has turned out to be a phenomenally important data set. So we go out multiple times in a summer. So we collect these things in the summer when they're in large swarms. We go out with our bongo net here. We lower it down to the bottom. It's about 180 meters where we go, pull it back up, and each side of this is used to collect 2 different samples. One we freeze for analysis, and I'll show you some of those data in a second, and the other we store in formalin so that we can then go through and identify and enumerate everything that's in the toe, or in the jar, as we say, that gets collected into this cod end here. So this is a super unquantitative way to look at how zooplankton hauls change over time. So you can see we go from 2001 all the way down to recent ones we just collected this summer. I'm going to show you some actual number data, but what I want you to get from this picture is just a sense of how variable these things are. Look at all the copepods, all these orange guys in 2011. This haul over here this year, 2015, not so great. Looks better in 2016, and you can just kind of see the variation from year to year. Well, we can actually quantify these. So what do the toes actually contain? And this is the sort of important data here. And what I'm showing you, the y-axis is giving you counts. So how many individuals are within the toe itself, or the contents that we put into the formalin jar. And we have 2 lines here. One is the dark orange line. Those are those Calanus stage C5, lots of oil, worth lots of energy in that dark orange line over time. And then blue is all the other species. And now I'll tell you it's mostly made up of Pseudocalanus and Centropages or Centropages, however you might pronounce that. And then along the x-axis here, we have toes in August and September going through time from when we started in 2006 to the last data we've analyzed, which is 2022. And what I want you to get out of this, and I'll show you some more information that will sort of support this, is that in the early part of our study, 06 to say 2010, 2011, there were lots of Calanus C5s in the samples. Like this orange line is pretty steady, first of all, starts to get a little more variable around 2010, but it's on top, right? There's more Calanus here than anything else. And the blue lines of what other species are there were pretty low. But as we move through time past 2010, 2012, and especially if we look at these recent years over here, these blue lines are absolutely spiking. And so Calanus is now not only not the dominant species anymore, and there's fewer of them. But these other species are really sort of taking over in our tows in the Bay of Fundy. You can kind of look at this if we put our data on a sort of average per year kind of plot. And the thing to keep in mind when you're talking about any kind of food source is you have to think about two components, not just quantity, how many are there, how many Fudgios are there, but quality, how big are those Fudgios and how much energy is in each one of them. And those two components are important for any predator, right? Because a change in one of those is going to impact how much energy a predator is going to be able to get out of a prey item. So here, if you notice, this is percentage of zooplankton. So how many animals in the jar, fraction-wise, are those Calanus C5s that are so valuable to the predators I pointed out at the beginning, like right whales, basking sharks, and herring. And over time, from 06 when we started to our last data point for this data set is 2021, there's fewer of them percentage-wise, but there's also fewer of them total. So there's less of them around. So that's the first sort of important thing to think about. Secondly, we're going to talk about quantity. And, oh, sorry, that was quantity. This is quality. So how big are they? And this is, you know, weight of each individual little tiny little C5, again, over time. And you can see there's some variation here, but there's generally kind of a decline. And what the point is that they were pretty big at the beginning of our study. And then following around 2012, they got really small. In some of the years, they've recovered a little bit, and now they're kind of in the mid-range. And to give you a sense of what this actually means, I'm using my sort of banana analogy, is that a banana is worth 89 calories. And how many little Calanus C5s do you need to get to the equivalent of a banana? In the early part of our study, you needed 36,000 C5s to make a banana. But in the later part of our study, you needed 54,000 C5s to make a banana. And so the value of each one of these has declined. And there are several reasons why we think this is happening, which I'm going to kind of tie in at the back end of the talk, but realize that quantity has gone down and quality has gone down. When you put these two things together and think about if you're a predator like a right whale, you're going to eat like a mouthful of copepods, right? So how much is in a mouthful? So if we define a mouthful as being a cubic meter of water, just to make life easy, how many joules, how much energy is in a cubic meter of copepods if you're a right whale predator? And again, variation here, lots of it. But what I want you to see is that these arrows, these blue arrows on the right, tell you the average sort of energy content of a cubic meter. And in the early part of the study, it was around 6,000 joules per cubic meter. And after 2012, it was more like 3,500. So a decline in energy value of 40%, which is a big change if you're a predator like a right whale who feeds, at least in the Bay of Fundy, exclusively on these things. So what does this mean? Predators are either going to get less energy or they're going to have to compensate by spending more time trying to find their food, or maybe both. And what this means is it will have impacts on their growth, their health, and their reproduction. And if you just think about sort of a time budget for an animal, if you used to spend half your day eating and now your energy value has gone down by 40%, you're really going to have to jack up the amount of time that you spend foraging. And so this is going to have impacts on all parts of the ecosystem. And I'll just show you a few pieces that are linked to this sort of change in copepod energy, overall energy density, quality, quantity since 2010, 2012, big decline. The first one is right whale sightings in the Bay of Fundy. So everyone's probably aware that right whales used to spend a lot of time in the Bay of Fundy. We would go out on an average day to the middle of the Bay of Fundy by the shipping lanes, where I showed you where we took those plankton samples, and you might see 40, 50, 60 whales in social groups. This is a study done by Record et al., published in 2019, who looked at right whale abundance or sightings per unit effort of survey data in 2004 to 2008 on the left and 2012 to 2016 on the right-hand side in 3 different habitats. And the one I want you to pay attention to is yellow, the Bay of Fundy. And then along the bottom here are just months of the year, showing you that we tend to have right whales, you know, in the summer months when they're feeding on those copepods. But the important thing here is that in this period, 2004 to 2008, there were many, many more right whales in the bay compared to, and the gray line is showing you sort of significant differences and means compared to the latter part of the study after 2012. So there were fewer right whales in the bay after the change in copepods, and those things have been connected by Record and many other authors. Herring landings is another question. So this is a DFO document that's a little messy, but it does show you what they are calling spawning biomass in the Bay of Fundy area, which is included in 4VWX, because herring is managed different than lobsters in different areas. But the line to pay attention to is this red 3-year moving average. And notice that there is a general decline over time, but really it starts to dip after 2015, 2016 or so. And there are obviously many, many factors involved in all of this, and I don't mean to simplify it down to one thing, but I will point out that Calanus copepods have been identified as an important part of herring diet. So if you're starting to see a decline in spawning herring, it's going to take a couple years for herring to get to spawning size, you might imagine that it might have something to do with a change in their prey base. No one's really done a good job of looking at that yet. And then last, thinking about larval lobsters, there's now some evidence that Calanus may well be an important prey item for larval lobsters. Carloni et al. Put together a paper that linked very closely post-larval lobster survival and lobster numbers with Calanus abundance. So good Calanus years, lots of post-larval lobsters, bad Calanus years, fewer of them. And there's actually a student at the University of Maine who's currently looking at stomach contents using eDNA to try and see how important Calanus is as a diet item for larval lobsters. I'll tell you, I don't know exactly what the lobster fishery looks like in Newfoundland, but around Grand Manan, our lobster landings peaked around 2015, 2016. The DFO survey data that tells us about biomass, this is sort of how they measure commercial biomass, had a peak roughly around 2018 and then a major decline. And one thing to keep in mind is that it takes 7 or so years for a larval lobster to reach market size. So you might imagine, and again, no one's looked at this in any detail yet, but you might imagine a decline after 2018 could reflect fewer larvae surviving sort of in the 2011, 2012 period, which is when we saw those changes to copepods. So these things kind of link together. So that gives you a sense of Calanus changes and how it might impact the Bay of Fundy ecosystem. So now we're going to talk a little bit about lobsters, and I apologize, I'm going quickly here. I'm trying to make up a little bit for time, but we've also been working with the lobster fishery to ask a couple of questions about lobsters. Where do lobsters with their eggs go? What kinds of temperatures do they choose? And have there been any changes in fecundity over time? And this is one of my favorite things that I do. This is an image from the boat that I fish on in, I don't know, mid-November. I love it because it's a little rough and it's a little challenging and my cell phone doesn't work out there. And so absolutely nobody can contact me, which I think is fantastic. So just to show you a little bit about lobster reproduction, I need to tell you a bit about their life cycle because it's quite long. And what happens is that a female lobster will become mature somewhere probably after 5, 6, 7 years, 4 years in some cases, and she will molt in the summer. And so that's, backing out of her shell. She's a little soft for a while the new shell kind of hardens up. And during that period when she's a little soft on the external portion of her body, that's the only time that she can mate. So males will mate with females right after molting. They'll deposit sort of a sperm packet with the female, and then she'll kind of harden up. And that following winter, her ovaries will develop their eggs internally. So when you look at the lobster, you can't really tell anything about her reproductive status because she just looks like a lobster. The following summer, she is going to extrude those. So she's going to spawn them. And as they pass out of her ovaries to the underside of her tail here and attached to these swimmerettes with a nice biological glue, that's when they get fertilized. So she stores the sperm, she then fertilizes them as she extrudes them and spends the whole next year with these embryos, which are now sort of independent of her energy source, but she's still got to keep them alive and make sure they're aerated. They develop over time. And then the following summer, they will hatch. So you can see this is a long time period. The little eggs hatch. They become planktonic larvae at the whim of the currents for about 2 weeks or so, molting numerous times, and then they'll settle out and start to grow into lobsters. And after her eggs hatch here, she's then going to molt again, and then we're all the way back at the beginning. So it's a kind of a long cycle. One of the interesting things about lobsters, female lobsters who are either carrying eggs or have been identified as carrying eggs, and I'll show that in a second, is that you can't land them. So fishermen, when they catch these lobsters, are not allowed to bring them in for sale. They're not even allowed to bring them into the wharf. So females carrying eggs, scientifically, we call them ovigerous, but the fishermen I work with called them berried because they look like they're carrying berries. In Maine, they call them eggers, same thing. But they can't be harvested. Even if this lobster has only one egg on the underside of her tail, she cannot be harvested. The other thing is that if you catch a female with eggs like this, you're supposed to cut a notch in her fourth uropod, like this lower circle right here, which will last through several molts. And it's meant as a conservation measure to protect reproductively successful and reproductively active females. So you can't land her if she's got even a hint of a notch in this uropod. And what they mean by hint of a notch is it has to be nicely filled in through several molts, and there have to be those little hairs on the end. And if those aren't there, you have to send her back. And so that means that these are perfect for a tagging study, because if you tag these animals and expect fishermen to help you sort of see where they go by catching them and reporting them again, you're not interfering with their catches at all because they can't land them anyway. So one of the things we've done is looked at a study that was done back in the 1980s by someone named Alan Campbell, who worked at the DFO office in St. Andrews, New Brunswick. And he came to Grand Manan in 1982. And he asked lobster fishers to put out tags on lobsters and then to recapture them and see where they went. And he tagged buried lobsters, so the ones with eggs. And then over time, he saw kind of where these lobsters were going. So he tagged 2,000 of them. He had a lot of recapture reports, better than we're doing, I'll tell you. But he also had a financial incentive. I think he offered $2 to everybody who reported a tagged lobster. And these are some of his returns. This 1759 around Grand Manan means 1,759 reports came from the waters right around Grand Manan. But he had some others go up a little bit to the sort of northern shore of the Bay of Fundy to New Brunswick. A couple went over to Nova Scotia, a few down into the Gulf of Maine. And his longest tag, at large, if you will, was 329 days. And so it seemed logical, since I was developing a working relationship with the lobster fishery anyway, to say, okay, 40 years later, what's different? And so we developed this absolutely super low-tech tagging program where I have yellow zip ties that essentially have a lobster identifier on it, 4-digit number, and my cell phone number. And we put out 5,000 of these since 2019, and we ask lobster fishers to report them. And we've had 1,300 reports, which is pretty good, from 900 individual lobsters. One of them caught 8 times, so you can see that some of them are definitely sort of familiar with traps and ready to go in there and eat some bait. I think this is a successful program. It was way more successful than I could imagine because this is sort of volunteer, because I make it really easy for the fishers to report a tag. We have a lot of help from folks. 164 different boats are reporting to us, mostly from New Brunswick, because I'm tagging right around Grand Manan, and so our Grand Manan fishers are going to catch them. But we've also had some reports from Nova Scotia and a bunch from Maine. I've done some outreach and some road trips to fisheries associations in other places. And this is, I think, an image of what makes this program easy for them. Literally all they do is, the easiest thing to do is to just take a picture of the lobster with the tag number in front of their plotter, and they text it to my phone. And then I get back to them within 24 hours and let them know where and when she was tagged. And they like the feedback because they can sort of, see where she came from, where she went. And like I always say, we're just sort of confirming scientifically what the fishermen already know in terms of lobster movement patterns. They know where they go, they know how to move their traps throughout the season, and this sort of just confirms what they're doing. So I'm not going to show you a lot of data here because my master's student at UNCW, Andriana Buckenberger, is currently working on this, but she sent me some lovely maps last week of some of her preliminary work. And so this map on the left shows you all of our initial tag deployments. Believe it or not, there's 5,000 points in here. And one of the things to think about with this study, of course, is that it's not fisheries independent, right? I'm tagging lobsters on lobster fishing vessels and then relying on lobster fishing vessels to catch them. So we're obviously not covering the whole habitat here because they don't put their traps everywhere. And so that is a limitation. But it's similar to the limitation that Campbell had 40 years ago. And so it's a similar kind of a bias. So here's where we put the tags. Here's all of our first initial recaptures, all of those 900. And you can see, sort of similar to what Campbell found in 1983, a lot of them stay around Grand Manan. Some of them go up to the North Shore here around New Brunswick, and some of them go into Nova Scotia and down into the Gulf of Maine. Looks fairly similar. So we've also learned that female lobsters are exceptionally independent. So here's an example of two lobsters who were tagged in the green blob in December. And then that following spring, one of them got recaptured very close to where it was tagged, and one of them was down here. So they're quite independent in where they go. We've also had a very long duration, 569 days, which was quite amazing. And the longest distance we've clocked is 160 nautical miles, looking quite similar to, again, what Campbell found with his work. So we're still working on this. As I mentioned, my undergrad student or my grad student is working on this stuff, and she's hoping to have it all wrapped up by sometime in the spring. Something else we've been doing, though, is putting thermal loggers on lobster traps. And so this shows you thermal loggers in a PVC housing to keep them safe from handling by crew members, but also being pinched by lobsters, which have very powerful claws. And we've been able to collect data from that. Lobster traps are amazing platforms of opportunity because what's going to go up and down through the water column multiple times during the week, and then you can sample the entire water column depending on what kind of a tag you have in there, traps, right? So it's perfect. We've also been putting thermal loggers on lobsters to ask the question of females with eggs, the V-notch females, what kinds of temperatures are they seeking out? Are they the same? Are they different? Because one of the things about lobsters that I probably should have mentioned is that they're phenomenally sensitive to temperature, and everything about their life cycle is dependent on temperature, from when they're going to molt, where they're going to travel to, how quickly their eggs will develop. And there is evidence that lobsters are sensitive to changes as small as 0.1 degrees Celsius from laboratory studies. So they're very sensitive to temperature. So what are they looking for? So we put these hobo data loggers on lobsters that are big enough. And when I proposed this study, we initially said we were going to put 50 out in the first year. And I thought, oh my goodness, if we get one of them back, it'll be absolutely amazing. We've deployed over 170 of them in the time we've been doing this, and we have 65 data records returned, which is beyond my wildest dreams that I ever thought we would get. And again, this is all due to cooperation from the fishing industry. We couldn't do it without them. Now, admittedly, some of these data records are short. You know, I put the tag out and then someone catches it like a day or two later, but some of them are quite long, and I'll give you some examples of what those look like. So here's an example from two lobsters who were tagged in December 2020 and recaptured in July 2021. And so we've kind of mapped these two on top of each other. And you'll see that the blue lobster spent time in water that was warmer than the green lobster. Now, one of the other limitations of these tags is that we don't know location, right? So I know where I released them and I know where they were caught, which were very similar, but I have no idea where either of these females has gone in the intervening period, and I also don't know how deep it is. So it does add a layer of complexity here. But you could say, oh, did the green lobster seek deeper water and that's why it's colder, or was she seeking colder water? It doesn't matter what her motivation is. It's that she spent time in colder water than the blue lobster did. Similarly, here's a sort of messy image of 4 different lobsters to start looking at things like, oh, is this a V-notch female, like the blue line here, or the green line over here, or a female with eggs, like this teal colored line or the brown one? And you can see that over the exact same time period, these lobsters experienced different temperatures. You can also see some interesting things we pick up on these data records, like right here, there was a warm water intrusion into the Bay of Fundy in March by a number of degrees. That was captured by a couple of our lobsters, but not this one. So it makes you wonder, she was obviously somewhere else. And then this is one of our more recent tags. I was quite blown away to get this one back since it was over a year long. And you can see that we can see what kinds of temperatures she was choosing over the course of more than a year. So you can get a lot of data out of these. And with 65 data records, we have a lot of analysis to work on that's still in the preliminary phase of things. One of the interesting things we have been able to determine from thermal loggers on traps, as I mentioned, is that it's warmer in the south than it is in the north. So here's Grand Manan Island again. Here's the shipping lanes. And then I had 2 fishers who were generous enough to put loggers on their traps, the purple one out here, close to where we do a lot of our zooplankton sampling, but further north, and the blue one down here, which is more into the Gulf of Maine proper. And over the course of not this past year, but the year before, you can see quite a difference between the purple trap, which is quite a bit colder, than the blue trap over the same amount of time. And these differences are even in the, amount of sort of 3 degrees. And remember, this is the bottom. So 3 degrees on the bottom is quite a bit of difference in an area that's not really that far apart. And then you might be asking, what are all these vertical lines? That's when the trap gets hauled and you see that it's exposed to external water temperatures. Some of the other things we've been looking at with lobsters, and this one's a bit disturbing, which follows into the next one, is changes in female size. And so this just gives you the distribution of how we measure female size, which is carapace length, which for lobsters is the distance from sort of behind the eyes to the end of the thorax, the carapace. That's how you measure female size. Minimum size to be landed, by the way, in the area I fish in is 82.5 millimeters, so like somewhere around here. And over time, while I've been fishing with the same captain in the same area, there's been an overall decline in average female size. That's this dotted line here, but you see that we're not getting those big females anymore since sort of prior to 2013. So we just don't see the big ones like we used to. The one thing that's very worrisome is changes in fecundity, and this is some work, and I should mention, since you're recording this and you're going to post it, a lot of this stuff hasn't been published yet, so please don't share it or cite it. This is one of my graduate students. We're still working on this manuscript with data we collected from this year. But in any event, this is calculated fecundity. So when I have a female with eggs, I measure the size of her egg mass and then collect some samples. And Jake, my student, went through and measured the size of the eggs for all of these females so that we could look at egg volume and then sort of estimate based on the size of the egg mass and a formula that was developed by Curry in 2010, how many eggs she has in her clutch. And that's what you see here. Carapace length, size of the female on the bottom here, and then estimated or calculated fecundity. And note that the open circles represent the years 2008 to 2011, and the closed circles represent the years that are more recent. And it should be fairly clear to you here that you'll see a couple of things. Again, we don't get the really big females anymore that we used to, that we saw in the open circles in the early part of the study. But more importantly, even at a given size, there are fewer eggs for an average female. And you can really see this if you plot average calculated fecundity for one size. Len, we just picked 100 millimeters carapace length. And if you look at average sort of calculated fecundity for females who are 100 millimeters in length from the early part of our study to the more recent years, this is a 70% decline. This is, I think, major cause for concern. Reasons for this could be a lot of things. Is the health of a female any different? Is she producing a different number of eggs? There's some theories that there are not enough big males anymore to fertilize these eggs because even though females who have eggs or V-notches can't be landed, males have no restrictions. So you can land all of those that you want as long as they're the minimum size. What I think is happening here is that because the effort has increased so much in the Bay of Fundy for lobster fishing, we have bigger boats. They're able to go out in weather that is worse than it was. And so they go out more often. And I think these lobsters are getting caught and caught and caught. And because the boats are so big and high now, when they get sent back overboard, these females may be, several meters off the water. And that's a big impact to their egg masses. And so I think that may be what's happening. And so the last question you might have asked me is, well, what about water temperatures? So if we go back to our initial map here, which is showing us the greater Gulf of Maine, and then our little Bay of Fundy, and there's Graham and Ann, there's a ton of data out there, and it gets supported every year with warmth in the Gulf of Maine, that the Gulf of Maine is one of the most rapidly warming bodies of water in the world. Tons of data. And this is just an example from a paper that Andy Pershing published in 2015, showing you this massive increase in water temperature in the Gulf of Maine. So what about the Bay of Fundy? Well, here's where the basking shark comes in, where, this is a little bit of a sort of right angle for you, but I'll tell you why this is important. So a number of years ago, we decided we were going to look at the ecology of basking sharks and their diving behavior to see where they go and how much of the bay they use in terms of the water column. Basking sharks are huge planktivorous fish, the second largest fish in the ocean, can get to be 11 meters. In our neck of the woods, they're a little bit smaller. They have a global distribution. They eat copepods as well. And we know practically nothing about them. We don't know how they reproduce, where they reproduce, the size of the pups, anything like that. And on top of that, with a global distribution, they seem to have very little genetic structure. So we don't really understand whether there are populations. But we have them in the Bay of Fundy, and we wanted to study their diving behavior. Just to give you a sense of size, these guys are big. If you've never seen a basking shark, and you are in a small boat and you see one, like this shot here of one of our research assistants from a number of years ago, these are really big fish. And then I'm hoping this is going to play for you, because the other thing they do is that they can breach out of the water, which when you're in a 16-foot boat or an 18-foot boat can be a little bit daunting, because there's this giant fish that has sort of the brain the size of a walnut that is jumping out of the water, and you have to wonder if they know you're there. And I'll just give you another look at this. This is from a friend of ours, Matt Lambert, who runs a whale watch business on the island. Okay, so we were putting these archival tags on these basking sharks. One of them shown here. It's got a dive recorder in it, and it is, we sort of attach it to the dorsal fin. It gets towed behind the shark for a couple of days. There's a magnesium link that degrades. The tag is buoyant, it floats to the surface, and then because it has a VHF tag and a satellite tag, we go out and recover it. That's a very, very fast way of talking about a very long process in terms of tagging, finding the shark, building the tag, getting the tag on, and then recovering it because it's an archival tag, so we have to get the data. We have to get the tag back to get the data. This gives you an idea of the kinds of diving behavior that we might get. This is from a shark we tagged in 2021. And this just shows you over the course of a couple of days, the water depth that the shark traveled to. And what we didn't realize when we started this study is that not only do our tags have depth sensors, right, pressure sensors to measure depth, like, oh, the shark went to the bottom of the Bay of Fundy because this is about as deep as it gets. There's also a temperature sensor on there. And so what this in effect means is that our tag sharks are acting very effectively, exactly like a CTD. So CTDs are conductivity, temperature, and depth instruments that you can deploy through the water column to get a sense of water temperatures throughout the water column. Because our sharks are diving up and down for a number of days, collecting depth, but also temperature data the whole time, we can also get thermal profiles of the Bay of Fundy from these sharks. Totally serendipitous. We had no idea this was going to happen. But anyway, what you end up with, this is a composite sort of plot that shows you multiple sharks tagged every year. These are the average temperatures, water temperature, at various depths for the years that we've tagged. And we do have data that goes back, I think we have 09 and 10 data too that we need to add to this, but I'll just tell you that 09 and 10 kind of sit right in here with sort of all the ones that are sort of blobbed together. So a couple of things to get out of this. Sea surface temperature is obviously quite different than what's at the bottom. Once you get below 100 meters, temperatures are pretty stable, so around the same. And then a couple of key points, 2012 was really hot. And it was recognized as an ocean heat wave in the Bay of Fundy, but also in the Gulf of Maine. It was really hot. And we're talking like a couple of degrees difference here, somewhere around, you know, 1.5 degrees, 2 degrees different to the average. So kind of keep that in mind. The second thing is, unlike the Gulf of Maine, we are not marching linearly towards an increase in temperature, because the year after we had 2012, when it was really hot, the next year was 2013, which is sort of in here. And this one, it went back to being the normal sort of coolish temperature. 2020, a fairly recent year, is the black line, kind of lumped in with all of the other ones. Our most recent year that we tagged sharks, we didn't tag any this year. Rachel, that will make you sad. The purple line here, you can see this one's pretty warm, and 2022 was shaping up to be a pretty warm year, but nothing like 2012. So think about 2012. These are data from lobster traps. And again, this is not a clean, super clean data set, but what it shows you is sort of winter data. So for each year, roughly December to June, in each case, the temperatures, monthly temperatures we got from the bottom from our thermal loggers on lobster traps. And again, I'll admit this is not a super clean data set, but what you see is from 2010, 2011, you know, it would get down to 4.5 degrees or so. It got a little warmer in 2016, but then it was cool again in 2017. And what you get out of this is that, yes, it looks like sort of fallish temperatures. So November, December are, you know, getting maybe a little bit warmer, but in the winter in the bottom, it's sort of not, there's a little bit of an increase here, you might imagine, but it's nothing like what we see in the Gulf of Maine. So I was telling you about that copepod sort of collapse that happened after 2010, 2012. And we can imagine a lot of reasons that that might happen, but we're not seeing this massive thermal shift in the Bay of Fundy other than 2012 being super hot. So what happened to those copepods? What I think is the case, and this is a really interesting study, and if you haven't seen it, you should look at it, by Aaron Mayer, Gutbrod, and colleagues from 2021, is what they noticed is a massive shift in sort of the physical oceanographic realm of the Gulf of Maine. And so over here on the left are the sort of the main currents that drive where water comes from and where it goes relative to the Gulf of Maine. And what they have noticed is that since around 2010, this Nova Scotia current here that comes down has started advecting into the Gulf of Maine. This is warm slope water. And it started to come into the Gulf of Maine and increased water temperatures there. And that's why we see all kinds of changes in the Gulf of Maine. And I think what's happened to our copepods up here in the Bay of Fundy, sort of by these like BF letters, is that we're not growing them in the Bay of Fundy. I think they're coming from the Gulf of Maine through ocean currents. And because the Gulf of Maine is a little warmer, if you think about an animal that's an ectotherm, what do you do when you increase the temperature? You have a Q10 effect, right? And if you're not a physiologist, what that really means is that you increase temperature, you increase metabolism. So they're going to mature at smaller sizes, and they're going to use up the energy reserves they have a lot faster. And we see that with this, like, how many copepods does it take to make a banana, right? They're smaller, there's less energy there. And I think that's the effect of where they're coming from in the Gulf of Maine. So more work sort of to be done on that one. Okay, a few summary notes here, and then I'll sort of wrap up, hopefully trying to get us a little bit back on time. In the Bay of Fundy, 2012 was an ocean heat wave, and since then things have changed. Copepods have become less predictable in terms of quantity, and other species are rising to become dominant, like I mentioned, pseudocalanus, but mostly Centropages or Centropages. A concurrent decline in right whale sightings. Remember, right whales are targeting C5s in their summer foraging areas. You've probably heard a lot of them have moved into the Gulf of St. Lawrence. We only saw one right whale last year. We didn't see any this year in the Bay of Fundy, so that's a major change. We've seen changes in the population of lobsters that we're seeing in the fishery. Not very many large females anymore, and fecundity is declining. And then anecdotally, I didn't kind of share a lot of this, but we've seen fewer humpback whales in recent years in the Bay of Fundy, which humpback whales are targeting herring. And I showed you that herring are sort of declining. So even though the Bay of Fundy is not following the exact same thermal trends that we're seeing in the larger Gulf of Maine below us, I think whatever's happening there is largely impacting what we're seeing in the Bay. And then a couple of take-home messages for you guys, your graduate students. So thinking about things in the future, a few pieces of advice. Long-term environmental monitoring, as I hope I've shown you, is absolutely crucial to understanding what is happening to any ecosystem over time, especially thinking about the effects of climate change. It's really hard to fund. Funding agencies want quick, immediate data, 1-year studies, maybe a 3-year study. So it's challenging to figure out how to fund this stuff and persuade people that the work needs to be done. Nothing happens in a vacuum. You need collaborators. We are no longer in sort of the single professor, single PI mindset where you can just get the work done by yourself. You need people with different expertise. For example, my lobster study, I understand lobsters, but not how to do GIS spatial analysis. And so my grad student needs a committee member in another department who has that expertise to help us figure this stuff out. And you also need people with different ideas, people from different backgrounds, different viewpoints, different life experiences to tell you about that. Which leads me to my next point. Traditional knowledge, community knowledge, however you want to define that, have enormous value if you think about people who've been on the land or on the water for years, and they're out there every day, and they're the ones who see the changes. And so listening to them and what they think and what they see and what they think are driving these changes can have enormous value and should be recognized, acknowledged, and used, the data. Be really creative with your funding. One great example of kind of how we've been creative anyway, at the research station, we can only get funding sources that are either provincial or Canadian federal or donations, maybe a little bit of support from industry. And so I managed to persuade the Department of Fisheries and Oceans to fund the lobster thermal study by arguing we needed to know about water temperatures to tie it into copepod predictions so that we'd have an understanding of right whale habitat. So there's a connect the dots, but they funded it. And so being creative with your funding is another thing to do. Every ecosystem is unique, so keep that in mind. And also, sometimes things come your way that you don't necessarily expect are going to come your way. Like, who knew that tagging basking sharks was going to give us oceanographic, like thermal profiles in the water column? We had no idea. And then my last piece of advice for you, which is the picture on the right-hand side here, is embrace the unexpected, but enjoy what you're doing. This is a picture of Koopman, my dog, who is asleep on the couch next to me here. But this little blob in the back is a fin whale. So he just watched a fin whale descend, and sort of that magic moment, I think about it all the time. So don't be afraid to love what you're doing and take in those little moments that are amazing. So with that, I'm going to thank a whole pile of people, but most importantly, the fishers that I work with. These folks are amazing. They know their business, they know the water, they know the animals. Funding agencies, and yes, we got funded by Big Spruce Brewing Company. That was fun. A bunch of local things, as well as provincial, some industry work, and again, some images of the folks that I work with. Couldn't do this without them. And so with that, I'm going to stop sharing. Hopefully there's still some people left. And I am happy to take any questions that you might have.
Great. Thank you, Heather. That was fantastic. Really appreciate you presenting for us today. With that, I'll open up the floor for questions. People can either unmute themselves and ask through the chat or here in the room, or they can directly text within the chat and I can read it out for you. Does anyone have any questions?
And I guess I have one.
Thanks. Yeah, I'll turn it around there.
It's kind of like not about the research that you're talking about, but you said you were from Guelph and that the research station that you are at was originally for Guelph, like Guelph students can come visit. I was wondering if they still do. I just finished my undergrad not too long ago at Guelph. So I was just wondering if you still work with students at Guelph, because I don't remember hearing about your research institute while I was there.
No, so I'll have to give you a little background. So it was started in the early 80s by a professor from Guelph, David Gaskin, a local fisherman, and a local hotel owner. And they started the research station, and they had it as a platform for grad students from Guelph to go out and do research. But the sort of kicker connection was the fisherman had a lobster boat and the hotel guy had a hotel. So they were also sort of doing whale watch tours with the lobster boat, and the grad students would serve as whale watch guides in that time.
Yeah.
So it was kind of an interesting model. But then the hotel guy backed out, and the fisherman no longer was doing the whale tours. And then the professor from Guelph, who was my master's advisor, passed away.
Oh,
And so
okay.
the connection with Guelph was sort of severed. And the research station became formally, it was always a Canadian nonprofit, but it sort of got detached from Guelph at that point. And then the fisherman on the island, the guy who had the boat, owned the house that we're in. And then he sort of deeded it to the research station. And so now we actually own the property that we're on as our own little non-independent self. Or sorry, independent self, nonprofit. So we don't have a direct connection to any university, but as Rachel can tell you, we do take summer researchers. So if you are interested in being a research assistant, we take a couple people every year. This year's research assistant was actually a fourth year student from Dalhousie. So he came out for the summer and spent some time with us, just like Rachel did, doing surveys, doing plankton tows, looking around a lot in the fog. Staring at the tide table and the flags at the ferry terminal across the street, seeing how windy it is. And so we do have students come and work with us. And also if you have a project that is, suitable for doing in the Bay of Fundy, we're always open to entertaining collaborations with students from other universities, but we don't have a formal connection to Guelph anymore. That sort of ended, well, Dave Gaskin died, I think in 96. So around there.
Yeah, before I was there for sure. Yeah, this is too bad.
Yeah.
I would love to come visit. It sounds like a really neat place, and you're doing very cool research.
Graham and Ann, probably like where you guys are, is either a love or hate it, right? It's a long ferry ride from the mainland. Not like you. We don't have a lot of the same, like there's no Starbucks, there's no McDonald's. And so some people like it. I love it. I got hooked the second I was there, but others don't. And so it's a very interesting place to do research. And also it is insular to the very word of that word, right? Getting into the community is a long slog. Like the amount of time, I have a great relationship with people now, but it's taken decades. And the amount of time that you literally are sitting at someone's kitchen table having coffee to build a relationship, And so it goes a long way to show you're sort of a human being first and a scientist second. And I've learned that after making some mistakes, but also just figuring out how to make inroads into a small island community.
Very interesting. Thank you. Yeah, it was really, seems like it's working out for you now.
It's totally working out for me now. And the reason I got into the whole lobster study was we used to do a lot of work with harbor porpoises, would get caught in these herring weirs. I don't know if you have weirs around Newfoundland. They're sort of like structures that herring feed into. They're close to shore. It's like big telephone poles and then a net around it. The herring would swim in, porpoises would swim in, they'd go to seine up the herring and the porpoises would smother. So we developed a program with the local herring weir fishermen to kind of get the porpoises out without killing them. And in the course of doing that, one of the guys I was working with is now my lobster fishing captain. And he was saying something about female lobsters. And I was saying, well, Neil, don't you think there should be a maximum size for females? Because don't bigger females produce more eggs and they're important and we should save them? And he said, what do you mean by a big female? And I was like, and of course, like geeky scientist, oh, I read the literature and, it's this size female. And he's like, that's like half the size of what we catch. You have no idea what you're talking about and you need to come fishing with me. So I did. And that started that whole project that just came about because I thought I knew what I was talking about and I didn't.
That's amazing.
Great. I had a question. So with the changing zooplankton community, have you been seeing new predator species coming through to the Bay of Fundy?
That's an interesting question. Last year was sort of like the year of the blue shark. We had so many blue sharks that, you never used to see them at all. And then last year you'd go out and see 20 in a day. This year we didn't see a single one. So I don't know what has happened there. And I don't know how that's connected to the zooplankton predators either, or the zooplankton community itself. Trying to think of other things that we've seen. We've sort of had years with lots of jellyfish, but that's not really related to the actual zooplankton. Didn't see any jellyfish this year. So I don't think we've seen an influx of a lot of unusual things. Although, again, no mola, not really very many molas, sunfish this year either.
So strange. Yeah, I guess it's hard to differentiate what factors are affecting the populations, right? Whether there's temperature or changing prey. Yeah, but
It's
of course
all
you're
of
doing-
that.
it's
Yeah.
everything all together. Yeah,
Yeah.
it's great. Does anybody else have any more questions in the chat? Gabby says that was an amazing presentation.
Thank you.
Oh, yeah, I'll turn this around.
Oh,
There's John in the
2015 was
back
such a
there.
poor year. Yeah, 2015 was a poor year, and 2013 was too, the year after the massive heat wave. So I really think that what happens in the Gulf of Maine, I used to think it was local Bay of Fundy temperatures that were influencing the sort of status of our copepods, and I don't think that anymore. And so I think having to go back now and see exactly what was happening in the Gulf of Maine in those years is probably one of the things we should do.
John there has a question in the back.
My question is somewhat related and to your point about the value of different time series. So you talked about Blue Bay, it's Blue Bay time series, that's the Gulf of Maine coastal temperature. Just wondered whether you've also considered the use of the Prince 5 DFO station that's off of Campbell Island at 100 meters, and whether that factors in and could extend some of the analyses back in time.
Yeah, that's an interesting point you make there, because we did have, I know about Prince 5. I know that for some of, the copepod study was done by a master's student. We wrote up the paper, we submitted it. It came back with some comments that we need to address and revise. And in that study, we had a couple of places where we were linking things to temperature and we were missing some temperature data. So we were using Prince 5 to sort of fill in a little bit of that. The water temperatures, so Prince 5 is located, closer to Campobello, around the Wolves, if you're familiar with the Bay of Fundy, and not in the basin where the copepods are. And the temperatures are quite different, and there's not an easy, direct, linear offset that we can use Prince 5 to directly fill in for where we are. So there's a little bit of complexity to that. Sort of telling you, and it's not surprising given the tides and the currents we have in the Bay of Fundy, that things are different depending on where you are there. But yeah, that's a good point. I mean, Prince 5's been going for quite some time. I think it's 90 meters actually.
Yeah, thanks so much. Great talk.
Great. Okay, if there's nothing else, I'll just wrap this up. Thanks again, Heather, very much for your presentation. And to everyone who joined us today for our first seminar this season. So our next seminar will be next Thursday, and it'll be at a different time than usual, it'll be 10 a.m. So that's on September 5th at 10 a.m. And it will only be online. And Dr. Flora Scorland will be presenting Identifying Casual Factors of Canadian Maritime Accidents Using a System Theoretic Approach. And like I said, this will just be virtual only. We do not have the room for that time. So I hope you can make it again for next week. Thanks again, and we'll see you all soon.
Thanks,
Cheers.
everyone.
Great.
So how are things going, Rachel?
Really good. Yeah, I'll just stop.